Here we have a diagram of RNA splicing. In RNA splicing, you will have the RNA transcript, which is made out of exons and introns. However, the introns do not need to be expressed. So therefore, inside the nucleus, you will have snRNPs made out of snRNAs and proteins, as well as other proteins. These two will come together and form a complex known as a spliceosome. The spliceosome will bind to the pre-mRNA through base pairing. Now, within the spliceosomes, the snRNPs will act as ribozymes and cleave out the unnecessary introns. The other proteins will then bind together the exons to form a continuous mRNA strand. After completing its purpose, the spliceosome will then be degraded into its components. Now, the mRNA strand will contain a 5' cap and a poly-A tail. The 5' cap made out of a modified guanine molecule and the poly-A tail made out of a series of adenine molecules. Now, depending on which of the genes in the RNA transcript are considered exons and which are considered introns, the proteins produced by the same RNA transcript may be different. This is known as alternative RNA splicing. So this is a diagram of the lac operon. The lac operon contains a promoter with a TATA box. It contains an operator and 3 genes, lac Z, which codes for beta-galactosidase, lac Y, which codes for permease, and lac A, which codes for transacetylase. The function of beta-galactosidase is that it breaks down lactose into glucose and galactose. The function of permease is that it is a transmembrane protein that allows the entry of lactose into the cell, and the function of transacetylase is still unclear. Now, within the cell, upstream from the lac operon, you will have lac I, which is the regulatory sequence for the operon. Lac I, when read, because there's a promoter, will produce an mRNA. This mRNA codes for an active repressor. This active repressor prevents RNA polymerase from reading the 3 genes and producing the 3 proteins. However, when lactose is present in the cell in high concentrations, it will act as an inducer and attach to the active repressor. And then it will change the active repressor's shape. It will change the active repressor's shape into its inactive form, thereby allowing RNA polymerase to read and produce these 3 proteins. Here we have a diagram of the trip operon. The trip operon is composed of a promoter, which contains a TATA box, an operator, and 5 genes. Generally speaking, within a cell, the trip operon is always on. This means RNA polymerase can bind to the promoter, read downstream, and form an mRNA strand. The mRNA strand is then read by ribosomes to produce 5 polypeptide subunits. These 5 polypeptide subunits form 3 proteins which break down a precursor into tryptophan, which is necessary for the cell to survive. Now, located upstream from the trip operon, you have a trip regulatory sequence. This regulatory sequence produces an mRNA strand, which produces an inactive repressor. When tryptophan levels are high in the cell due to the 5 polypeptide subunits, tryptophan will bind to the inactive repressor, causing the inactive repressor to change shape and become an active repressor. The active repressor will then bind to the operator. What this does is it prevents RNA polymerase from binding to the promoter. Therefore, it is then negative feedback because it prevents RNA polymerase from producing 5 polypeptide subunits. In this diagram, we have distal control elements located on an enhancer on a strand of DNA. Activators will bind to the enhancer. Next, DNA bending protein will cause the DNA strand to bend over, and a group of mediator proteins will enter in and bind to the activators. Next, General transcription factors will bind to the group of mediator proteins, and RNA polymerase will bind to the general transcription factors over the promoter, which has a TATA box. What this does is it forms a transcription initiation complex, which increases the speed of the transcription.